kind. My name is Canan. I am a faculty at MIT, and in my research group, which is called Conformable Decoders, we are trying to convert the patterns of nature and the human body into beneficial signals and energy. And in this regard, conformable means flexible, stretchable, mechanically adaptive structures, and decoders are the devices that we create to decode the messages that we have coming from our human body parts. So we believe we live in an ocean of physical patterns, heart rates, respiration, neuronal activity, and so on. And all these patterns contain information in coded ways. And we would like to decode these messages through our sophisticated devices and understand what body parts are really trying to tell us. So since throughout my journey, academic journey, I have been decoding, conformably decoding out of the body, mostly wearable devices on skin or attachable devices, mainly on your personal garments like bra, underwear, t-shirt, and so on. And as well as we have been decoding in the body through either ingestible devices that you take orally or implantable devices in the deep brain. So this is a quick snapshot of the devices that we have done. Human body, we believe, is magical. Eyes, nose, skin, they all talk to us. And we need conformable devices with micro-fabricated features, which can allow us to understand how the body parts are functioning and communicating with each other. Those devices can be rolled like a carpet and placed in a dissolvable capsule, and they can travel all the way from mouth to the stomach, and the dissolvable capsules open up and sticks to the stomach lining, kind of a Fitbit for your stomach, and tells how much food intake and out that you have. Also, these devices can be implanted on moving organs like heart and diaphragm, and harness mechanical energy into electrical energy. And first time ever in literature, we process 8-inch wafer piezoelectric devices on silicon wafer, and then decouple them from their mother substrate and laminate on facial skins like your cheek. And you can make thousands of messages through facial gestures, especially for people who have speech disorders like ALS. And these devices are such tiny, and you can even camouflage them with makeup products so that they will be more socially acceptable. Or these devices can be a part of your personal garments, can be washed multiple times, and measure different types of vital signs to find the signatures of different types of diseases. So right now, actually, the shift that we have in our group from micro-scale device level to the system level, and you may ask, why do you do all these kinds of different types of devices? So the idea that we have in our lab, we would like to explore the human body with a holistic approach. And collectively, we believe tissue across the human body create a systemic interaction. And when you interface with one end, you can actually modulate the other end through a specific pattern. For instance, imagine you have an implantable device which goes inside a deep brain, which will be mainly the scope of my talk today. And this device can stimulate your neuronal activity through chemical stimulation, and at the same time read out neuronal potential to create explosive system. And at the same time, you may have an ingestible device which can monitor the motility in your GI tract, and at the same time, a wearable device on your knee as a patch, which can monitor your gait quality. And by having 3 different types of device formats for targeting the 3 different organs and understanding the communications in between, connections and communications in between, we can really understand the underlying mechanisms of neurological disorders such as Parkinson's disease. So instead of stimulating the brain with chemical stimulation blindly, we can understand and trigger the motility maybe, and at the same time understand how the gait quality is occurring throughout this process. So this is the big picture that we have in our group that we are currently pursuing. So, and we do all these devices in our clean room dedicated for our all research at the Media Lab. And this, we call this lab yellow box. It's literally yellow because of the UV sensitive polymers that we use. And at the same time, this is how we look like in the lab. Nobody can recognize who's who, which is perfect, so that we can do as crazy science as possible without being judged in the lab. And this is a code that I found online, which I found very valuable, which captures actually today's our understanding about the brain. Our own brain is equivalent to the Milky Way, and what has been understood so far is at the scale of the solar cell. It's because not only the brain is a very complicated organ, it's also because we have limited tools to understand how the brain really functions. And in our group, to tackle this problem, we are creating implantable devices which are thinner than your hair fiber, thanks to the microfabrication techniques that we use. And we call them MINDS, minimally invasive drug delivery systems, a device for chronic treatment of the circuit disorders. So what we do, we pass the skull and we read out the neuronal activity. And when the neuronal activity is not good enough, then we normalize it through chemical stimulation and have a normalized behavior for different types of neurological disorders. And this is how the device looks like. We have a needle type device, all in a stainless steel backbone. So we don't need additional guide tube to have the device to be penetrated inside the brain with a targeted location. And at the same time, we have remotely controllable micropumps, which we can house two different distinctive drugs inside the brain to understand the behavioral activities. So here how the device looks like. We do have a device backbone which is made of stainless steel because it's a biocompatible. We lower the sound a little bit. Okay. So, and the tip of the device has a borosilicate tip aligner to align all the tiny components that we have, like a tungsten electrode to read out the neuronal potential, and borosilicate channels for drug infusions, and microfabricated polymer template to in-house everything in a stainless steel device backbone. And the borosilicate tip aligner, as I said, provides a protective confined region for the device, and tungsten electrode for neuronal activity for potential feedback, and this microchannels for drug infusion. And thanks to the microfabrication, we can make these devices as large as 10 centimeter for large animals like macaques, and small as 1 centimeter for the small animal models like mice or rodents. And through the path structure, we show that we can have a controllable volumized structures inside the brain with no backflow or diffusion, which is extremely important for neurological applications. And specifically for this experiment, we target a special location called substantia nigra in animal model brain. And by just simply using our code sent through our computer, we can actually make the animal to turn right or left, run and stop, by creating the hemi-Parkinsonian state through a muscimol infusion. And not only smaller animal models, this device can also be implanted on larger animal models like macaques. And we can infuse different types of drugs like muscimol or ACSF to wash out the effects of the muscimol, to change the firing modality inside the brain. And this is the first reported system which allows two distinctive drug administration with picoliters of amounts, which are 6 orders of magnitude lower that you take orally or intravenously, which is dramatically decreases systemic toxicity and at the same time increase the behavioral changes within sub-seconds. So, of course, there are amazing scientists all around the world who target the brain for several reasons, because it's a heterogeneous and we know so little about it. And there are different structures like microelectrodes, microfluidic channels, and so on. And the need here, we need small volume modulation, especially for the deep brain regions, and smaller probes. And at the same time, we would like to have a larger aspect ratio, at least hundreds, so that with pinpoint accuracy, we can go inside the deep brain without creating less harm or tissue scar while doing different types of interventions. So, I'm sorry. So here is how the device looks like. We have a SEM image, and as you can see, the minimally invasive neuronal system that we have has a diameter of 200 micrometer with an aspect ratio of 500, and tested in smaller and larger animal models. And the tungsten electrodes that we have, as you can see here, at the tip, we have the exposed region like a pen, and rest of the device is covered, instead of a polymer, a dielectric stack layer made of silicon dioxide, aluminum nitride, and silicon dioxide, all deposited with aluminum atomic layer deposition, like layer by layer. So it's extremely thin, which makes our device to be as small as possible, and how our tungsten electrode to go through the borosilicate tip aligner that we design. And not only that, we try to capitalize on our microfabrication skills that we develop over years. And what we use a silicon wafer, and on top of the silicon wafer, we use a sacrificial layer, which is called PMMA, which can be dissolved in hot acetone, and then create our PI template throughout different sets of microfabrication steps, and align all the components with an aligner, and then encase everything with epoxy, and then immerse everything in hot acetone so that the PMMA, which is a sacrificial layer, can dissolve away, and the device is simply swimming inside the hot acetone. Then we use a vacuum tweezer to hold from one end of the device, and then align it with stainless steel, and suck from the other end of the stainless steel to align everything through the backbone of the device. And we can create different types of structures with a tip aligner, very tiny at the tip, and as well as very long, depending on where in the brain that you would like to go and read the neuronal application, neuronal potential, and at the same time infuse multiple drugs at the side of the electrode. So these are some of the confocal fluorescence microscopy analysis implanted for 8 weeks in an animal model, where we show different types of immunohistorical staining. And as seen very clearly, though no significant tissue glosses appeared over time, and only the response was limited at the immediate surrounding of the stainless steel tube that we have, which is a great way of confirming that the device works with no issue inside the brain. We also had a follow-up study. This is published in Science Translational Medicine, and another one in PNS, where we show that the device can be implanted in animal model up to 2 years, and there is no clogging, and we can still infuse drugs and read out the neuronal potential with no degradation over time. So here, the next thing that we did to do in vitro characterization on with the micropumps, controllable micropumps that we have, and create a structure for researchers with a framework, how much drug infusion they can offer with this kind of interfaces, and how the diffusion is limited around the bolus structures that we create in the deep brain. The beauty of it is the internal resistance of our borosilicate channels are 20 micron, which creates a huge internal resistance. So when you make the pump off, there is no enough pressure for the fluidics to infuse further inside the deep brain. That's why we have a very controllable micro bolus areas, volumes within the structure that we are interested. And after confirming different types of drug infusion rates through syringe pumps in agarose and syringe pump in brain and the microcontrollable pump that we have in the brain, we did make a correlation and find the best regimen for our applications for the paper and for the behavioral studies that we studied further. And I'll just pass through this detail graphs, but technically what we showed here through PAD in vivo imaging given that our device is MRI compatible, we can show the bolus volume is perfectly repeatable and as well as controllable for smaller regions inside the brain. And the next experiment that we did, acute electrophysiological recording through tetrode electrode that we have with 4 tungsten electrode created as a bundle that you create like a yarn and place in the mind's interface that we have. And here what we did, we infuse a silane and show that there is no firing activity because it's an inert medicine or solution per se. And then we infuse muscimol and another sets of muscimol to show how the firing activity is diminishing over time. And at the same time, check the different peak values for different regions and check the mean of the action potentials from the same recorded units, which shows a stable configuration over time, confirming the unit that we recorded is the same unit, which means there is no major deflection, even though the device is placed in the animal and the animal is free to move thanks to the wireless neural stimulation technology that we have. And again, as shown in the picture that video that I showed earlier, we have the camera system on the top of the chamber, and we place the device surgically inside the animal brain and the pumps under the shoulder of the animal. And there is no tethering, no infusion cannulas or electrical cables outside. Everything is within the body of the animal. And we show that by infusing the unilateral delivery of muscimol, we can create a preferential rotation by reflecting in the hemi-Parkinsonian state and in the animal, which shows that we can change the behavioral activity within sub-seconds, given that we are inside the brain with our neuronal interface. And in this configuration, this time we switched gears to the larger animal models and tried our device on a site, on a macaque structure, where we infuse ACSF and muscimol, and at the same time did multiple infusions, ACSF, muscimol, ACSF in the same location, and measure the neuronal activity over time and see if we can recover the reduced activity by infusing another sets of ACS, which is an inert solution inside the brain. So the idea is, can we increase the firing activity or decrease the firing activity with distinctive drug administration in the same location and by reading the neuronal activity at the side of the infusion cannula? So in a summary, with this study, we created a minimally invasive, customizable, low bending stiffness device with high aspect ratio. And also, again, thanks to the microfabrication, we can adjust the number of the channels. Right now, for instance, in addition to having a micro infusion cannula, we are also inserting an ultrasound stimulation and as well as a pH sensor and pressure sensor, so that we can see how the tissue, brain tissue, is responding to different types of modulations inside the brain. And also this technology allows us to do on-demand selective drug delivery on the brain structures at the order of 1 millimeter cubic area volume. And also this is kind of a new frame way to deliver different types of neurostimulation inside the brain, like chemical or through optogenetic and electricity, and not also only limited to brain, to other types of organs like stem cell infusions in different internal organs such as heart. So with this, I think I'm on time. We believe that today's medicine is a pajama type, like you can wear your partner's, mom's, dad's pajama. It doesn't need to fit on you. But we believe you need to make a suit type medicine, personalized medicine, through advanced unusual engineering, in which the electronic devices, conformable devices, can have intimate integration with the target tissue and can really translate the biological language into electrical language for us to make a better personalized treatments for a variety range of diseases that we tackle every single day. And with this, I acknowledge my amazing mentors and group students, as well as our funding agencies. Thank you.
Great, thank you so much.
Okay, I think I see the questions. Okay, one private question. So maybe I may not hear. So if you have any question, if you can
Shannon,
say
are you able
out
to
better.
hear us in the room?
Okay.
Yes, okay, we're gonna open it up to audience questions and then we'll also take questions from the chat.
Okay.
The first question from the room.
Hi, John. Thank you very much for your talk. I really enjoyed it. This is Vamshi speaking. You mentioned that in the live experiments, your solution was completely wireless. Was that true, or were there like wires coming off of the Minds device? And regarding like a closed loop setup, is there potential to do that sort of computation on the device itself, or will that also be like streamed off the neural response sort of process offline, and then that will determine drug delivery?
Let me see if I really understood your question, because there is some echoing between. I think your first question was related with the optogenetics and virus infusion, right, through the channel. Am I right?
Correct, yes.
Yes. So one of my mentors is here at Boyden, and we are exploring with him to see how the infusion cannulas would be compatible with the targeted virus injection through our interfaces. So it's early to really say that it works. We are still exploring about it. But one thing that I could say in it, with respect to the additional channels that we are trying to add in our existing interface, is ultrasound stimulation inside the brain. I think right before Professor Vitale, there was an amazing speaker who talked about the ultrasound stimulation. So we are trying to do a similar work, but instead of powering, this time mechanical stimulation through ultrasonography as being one of the channels of our interfaces. So I also know one of our devices being used by currently investigated by another research group, where they try to infuse stem cells to the defunctioning heart tissue. So hopefully that will be published soon, and you can see how the device is instrumental in that regard. Hope this answered your question. And I'm so sorry, but I think I missed the second part of your question. I did not hear it well. It was related with computational aspect of the device. Am I right?
The question was whether the computation was on device, or whether.
Oh, yeah. Yeah. Okay. So in terms of the drug infusion, it's a wirelessly working. It's a pump that we actually purchased off the shelf, but we hacked it based on our needs by changing the infusion cannula and diameter and the modulus value of itself, so that we can adjust and control the internal resistance. So when it comes to the infusion, all the computation is through the device, through the pump, with simple codes that we send through our computers. When it comes to the neurological potential reading, we have a DAC system that we can hook the device in acute setting and read out the potential, as I show in the acute electrophysiological sensing, and we can do the post-processing afterwards.
Okay, thank you so much, John.
My pleasure.
Let's thank our speakers. Let's thank all 6.
Thank you very much.
I know this is my research area, but I was still so inspired this morning to think about and potentially work on completely new ideas that I'm not currently working on. I hope some of the students in the audience, whether online or here in the room, felt the same way. So thank you so much for the amazing talks this morning.